Hello everyone, today we're going to be going over how to use the gram stain technique in the lab. The gram stain uses four main stains. We're going to be using our crystal violet, our iodine, our safranin, our decolorizer, which in this case is acetone, and other materials we're also going to use is our bacteria sample, a slide for the bacteria, an inoculating loop, and a torch. Let's get started. So to start, the first thing we're going to do is get our water bottle and place a drop or two on our slide. The next thing we're going to want to do is sterilize our loop, and to do that we're going to grab our torch and flame the inoculating loop until all of it is bright red. That ensures that if there is any bacteria on the loop, it is killed off before we get our sample, so that our loop is nice and sterile when we grab our sample. You're going to want to wait about 5 to 10 seconds for the loop to cool down before you grab your bacteria sample. Otherwise you might damage your bacteria sample and it will result in a bad morphology as you're looking at the bacteria under the microscope. So I'm going to take my flaming loop, just grab a very small sample of the bacteria. Remember, less is more, and then I'm going to mix it in the water, diluting the bacteria. After that, you're going to let your slide dry. You can either do that through air, through the torch, or the preferred method is to put it on the hot plate, which is a much quicker method. Once the slide is completely dry and heat fixed, we can begin by adding our crystal violet as the first stain to our slide. The crystal violet acts as the primary stain, and we are going to pour that on the slide so that the sample is completely covered, and we're going to allow that and let it sit for about a minute. After the minute has passed, you can take the water and rinse the crystal violet off, and then move on to your next stain. The next step in the process is going to be to add our mordant, which in this case is iodine. You're going to do the same process that you did with the crystal violet. Apply the iodine so that it's completely covering the slide and allow it to sit for about a minute. And then after that minute is passed, you can go ahead and rinse the slide just how you did with the crystal violet with water, ensuring that all the iodine is off of the slide, and then move on to the next step. Now, after the iodine has been removed from the slide, the next step is going to be to add our decolorizer, which is acetone. So there's a couple of different ways you can do with the acetone. The first method is just applying the acetone directly on the slide and letting it sit for about 15 to 30 seconds. You don't need to do it as long as you did the other stains. The other method you can use is just a wash method, where you're constantly washing the slide with acetone and waiting until the drip runs clear. So you guys can utilize either way and find out what works best for you. And then after you're done with the decolorizer, we're going to move on to the final stain. The final stain we're going to be using today is the safranin. So we're going to be doing the same thing we've been doing with the other stains. Place the safranin on top of the side, and we're going to let that sit for about 45 to 60 seconds. After that 45 to 60 seconds has passed, we can go ahead and wash away the safranin with our water. And now all of the stains are completed. So the next steps would be to get rid of that residual safranin, and you can go ahead and blot the slide dry with some lens paper, and now your sample is good to go and you can put it under the microscope. So those are the steps for the gram stain. I hope this video was helpful in showing you how to do the gram stain in laboratory. Your slide is now complete and ready for you to look under the microscope, and I hope that you can use this video to fall back on throughout the semester. Thank you.